So here we see the way that two different disaccharides can be formed. The top one we already talked about. That's the formation of maltose with um, two molecules of alpha glucose. Okay, so they don't specify here, but this is alpha glucose. And we'll talk about beta glucose in a little while. I'll explain to you what that is. So if you take those two alpha glucoses, anyway, pull out of water, get the water out of there, dehydration synthesis. This oxygen then ends up being the connector, forms that 1,4 glycosidic linkage because we're dealing with carbon 1 and we're dealing with carbon 4. So that makes sense. And glycosidic just means sugar. The disaccharide that forms is maltose. All right. The other one that's shown down below here you do not need to memorize. So maltose you should be able to draw from memory. You should be able to figure out the two glucoses, the way they link. You should know that the hydroxyls go down, up, down, down. Well, this used to be a hydroxyl. This one is down, up, down, down. For the formation of sucrose, which is basically table sugar, that's one of the most commonly used sugar in food. You don't need to memorize this. You don't need to know it. But I just kind of want you to know the idea. And I do want you to know that sucrose is glucose and fructose combined together, but you don't need to know the structure. So notice that fructose is also a hexose. It also has 6 carbons, but 2 of them stick out of the ring. So the ring is only a pentagon. So essentially, fructose has this shape. The oxygen is here in the middle. Then it's a pentagon like this with carbons, and then one of the carbons sticks up and the other one sticks down like this. All right, once again, you got dehydration synthesis. You got the OH and the H forming water, just like before. But in this case, it's a 1,2 glycosidic linkage, and I'm not too worried about this. But just notice that it's not 1,4 anymore, and that's how we form sucrose. All right, well, if you link a whole bunch of glucoses, alpha glucoses together, plants do this all the time. After photosynthesis, after the glucose is produced in photosynthesis, the plant cells will link together those glucoses to store that sugar for energy for later, and they form something that's called amylose, and commonly that's known as starch. Okay, so there's 2 forms, amylose, amylopectin. We're not going to worry about it. One of them has more branches on it. Starch tends to be less branched, but that is a form of sugar storage that's used in plants. And if you know that a lot of types of plants have a lot of starch in them, like corn or potatoes and things like that. Grains do. So anyway, the polysaccharide made out of alpha glucose that's in plants is starch. Animals kind of do this also, a similar process, except they form glycogen. And glycogen is also long chains of alpha glucose, just like starch, except it's a little bit more branched. So you notice a lot more branching going on like this. And then animal cells basically create the glycogen to store as ready-to-go energy, and it's stored in muscle cells because muscles use a lot of glucose. They need a lot of energy, so it's ready for them there. And it's stored in the liver for fairly quick energy that's available for bursts of activity and things like that. Fat is another way that animals store energy, but it's maybe a little bit slower to access. All right, so that's the difference between starch and glycogen. There's also beta glucose, which we haven't really talked about at all. So beta glucose would look like this. You would get, basically you start the same way as alpha glucose. You put the oxygen at top right. You draw your good old hexagon, just like always. It's good to practice these, so you might want to draw this also. I'll put my OH here, put the hydrogens here. Sometimes we abbreviate this as CH2OH. It's the same thing. It's just shorter. Down, up, down. And now because it's beta, it's going to go up over here. So that's the difference. That's the only difference between alpha and beta glucose, is that the last hydroxyl, or maybe the first one, because this is carbon 1, goes up instead of down. Obviously, we have to fill all the rest in with hydrogens because we got to maintain that 1-to-1 carbon to water proportion. And we obviously have to come up with C6H12 12O6, because it is glucose after all. It's just a different isomer of glucose. Alpha and beta are different isomers of glucose. Same formula, slightly different structure. You might say, does this make a big difference to have the hydroxyl pointing up? And the answer is yes, it makes a gigantic difference, because this is basically the difference between sugar and, let's say, wood or paper. Because beta glucoses, when you link them together, they will form something that we call cellulose. Oops, that's too many L's. Cellulose. That looks too ugly. I need to fix that. Give me a second here. Oh, I deleted the whole thing. Okay, never mind. Let me just see if I can. Nope. Oh, this is cool. So the decision I have to make now is, do I... Oh, I can't erase. Do I start the whole video over and fix this? Nope. Just don't care. I'm just going to leave it as... Spelled it wrong again with the L. So cellulose. All right. Cellulose is the stuff that's in wood, paper, cotton, stuff like that, which is really tough and obviously not something that we can eat. So makes a huge difference. So the difference between a candy bar and a baseball bat is this tiny little difference in the way the hydroxyl points, whether it's up or down, which is crazy. When these link together to form the cellulose, here's what happens. So you got your glucoses connecting, and what's going to happen is they're going to connect like this. So one of them will point up, where that carbon 6 points up. Here it points down. Then it points up again. And this is different from starch, because in starch what's going to happen is all of them point up. And here they alternate. You might say, well, what's the big deal? Well, the big deal is that when this comes around and goes back over here again, what's going to happen is these molecules can form hydrogen bonds with each other. All these glucoses can form hydrogen bonds, and what's going to happen is a whole bunch of, we know each hydrogen bond is kind of weak, but a whole lot of these hydrogen bonds will end up being very, very strong, and that's what's going to happen. And this does not happen with starch and alpha glucose. No hydrogen bonds form. But here it forms these sheets that are really strong, and what makes it even better is the sheets roll up into these fibers, like cylinders, okay? And the fibers are very, very strong because of all the hydrogen bonding that's happening between the beta glucose molecules. All right, if I go back for just a second here, I should point out that the alpha glucose molecules in glycogen and starch form helices. Okay, so that's shown here, but I didn't really point it out before. They form these helices, which aren't as strong at all. I mean, they have some strength to them, but they're pretty easy to break. As you know, starch and those types of things are not very strong. Like you wouldn't want to make clothing or houses out of starch. It would just break apart real easily, and it would dissolve in water. Whereas beta glucose does not form helices. Instead, it forms the sheets, which then roll up into these sort of fibers, kind of like a cylinder that's made out of a rolled up sheet, and it gets really, really strong. And that's the wood fibers then that we end up using for lumber and to make paper. That's the fibers in cotton, etc., etc. All right, this is just a nicer picture of what I was just showing you. So alpha glucose here, we got this hydroxyl on carbon 1 pointing down. Here it's pointing up. Here all of these are pointing in the same direction. They just have them flipped over, but they're all in the same direction. So that's starch. That's the weak one. That's the one we can digest. We can eat this. Here we got one going down. We got one going up, down, up. They alternate. So that's cellulose because it's beta glucose. And this one is alpha glucose. And this is the one that's going to form the sheets and the fibers. So this picture kind of shows that. It shows how there's linkages to form these sheets here, the hydrogen bonds that form, and then these all twist together to make these fibers like that, which is then basically the fiber in the cell wall. Oh, by the way, I should mention cellulose makes up the cell wall of the plant, and that's why plant cells are so strong and rigid, is because they have that strong cellulose in them. Don't forget plant cells also have a cell membrane in here. There's a cell membrane, but then there's a strong cell wall all around, made out of cellulose and some other stuff too, but that's what gives plants their strength. All right, so we have this table here that needs to be filled out. Basically, I'm looking for monomer, I'm looking for where it comes from, what is the structure, can we eat it, and then what's it good for. So starch, the monomer clearly is, yep, alpha glucose. Glycogen, it's also alpha glucose, because both of those are digestible by us. And then cellulose, it's beta glucose. All right, the source of starch is plants. Glycogen is plants. Oh, I'm sorry, glycogen is animals, my goodness. And then cellulose is plants. Three-dimensional structure is helices. Glycogen is branched helices. And then cellulose is sheets and fibers. Digestible? Well, yes, of course. And then glycogen, yes. It's why we make it and store it, because we can break it down again for energy. And then cellulose is no, we cannot digest it. This is basically what fiber is in food. So if you're eating like high fiber cereal that has a bunch of cellulose in it that we cannot digest, you might say, well, that's bad. And it's actually, in limited amounts, it's good for us because it helps the digestive system become more efficient. It moves things through the digestive system with a little more urgency, so that things don't get bogged down in there, which could lead to medical issues, constipation, and other serious issues. Uses, so starch, it's in food, like flour, cornstarch, potatoes, all of that kind of stuff. Glycogen is stored energy in the liver or in muscles. Oh, and by the way, starch for plants also is energy storage, of course, but then we use it for food. We take advantage of that. And then cellulose uses and other info, it's in wood, so any kind of wooden structure like houses, baseball bats, stuff like that. Paper, cotton, so clothing. And then obviously for the plant, it's the cell wall. It gives it strength. It's what makes the plant strong and what makes wooden trees very rigid and strong. And then finally, this is kind of a minor point. There are carbohydrates that have other elements in them. So far, all the carbohydrates we talked about had carbon, hydrogen, and oxygen, and that's it. It's like Harry Potter's girlfriend, Cho. There's one that has nitrogen in it also, which is weird, and that's chitin. By the way, it is pronounced chitin, not any other way. So please pronounce it correctly. Chitin is a tough, cellulose-like polysaccharide that is found in exoskeletons of arthropods, like insects and crustaceans, crayfish and lobsters and things like that. And it is used as a structural or protective tough shell. Sometimes humans use chitin for making sutures, surgical thread, and I've also heard of chitin being used in violins. Not in violins, but as for the varnish for violins, that it makes the outer lacquer coating, because it works really well and gives it a good sound.